Hello friends, welcome to BMH Learning. This video is about expressed sequence tags. In molecular biology, an expressed sequence tag is a short subsequence of a cDNA sequence, usually 200 to 500 nucleotides long. ESTs may be used to identify gene transcripts and are really of a great use in gene sequence determination. ESTs are generated by sequencing either one or both ends of an expressed gene. Or in other words, ESTs are fragments of mRNA sequences derived through single sequencing reactions performed on randomly selected clones from cDNA libraries. From cDNAs to ESTs. Once cDNA representing an expressed gene has been isolated, scientists can then sequence a few hundred nucleotides from either end of the molecule to create two different kinds of ESTs. Sequencing only the beginning portion of the cDNA produces what is called a 5' EST. A 5' EST is obtained from the portion of a transcript that usually codes for a protein. These regions tend to be conserved across species and do not change much within a gene family. Sequencing the ending portion of the cDNA molecule produces what is called a 3' EST. As these ESTs are generated from the 3' end of a transcript, they are likely to fall within non-coding or untranslated regions, and therefore tend to exhibit less cross-species conservation than do coding sequences. ESTs as gene discovery resources. Because ESTs represent a copy of just the interesting part of a genome, so they have been proved as powerful tools in the hunt for genes involved in hereditary diseases. ESTs also have a number of practical advantages in that their sequences can be generated rapidly and inexpensively. That is, only one sequencing experiment is needed per each cDNA generated, and they do not have to be checked for sequencing errors, as mistakes do not prevent identification of gene from which the EST was derived. Unigene, a non-redundant set of gene-oriented clusters. Because a gene can be expressed as mRNA many times, so ESTs ultimately derived from this mRNA may be redundant. That is, there may be many identical or similar copies of the same EST. Such redundancy and overlap means that when someone searches database EST for a particular EST, they may retrieve a long list of tags, many of which may represent the same gene. Searching through all these identical ESTs can be very time-consuming. To resolve the redundancy and overlap problem, NCBI investigators developed the Unigene database. This was all. Thanks for watching.